So next one is meiosis 2. Actually, after completion of meiosis 1, the cell will enter into a small gap period called as interkinesis, where it is considered to be a resting stage. There is no duplication of the DNA molecule, no change in the chromosomes, just it will be in the resting stage, and once again it will enter into meiosis 2. So here in the meiosis 2, so as like we had discussed that 4 stages are there. One is called as prophase 2. In the telophase 1, we have seen that decondensation of the chromosomes will take place, so that the chromatin will be formed. That chromatin once again during prophase 2, the chromatin once again will be undergo condensation, and it forms into chromosomes. Then the Golgi complex, endoplasmic reticulum will disappear. The nuclear membrane and the nucleolus will also disappear during this stage. So we can see disorganization of the nuclear membrane like this, and nucleolus will also gradually disorganize, and the chromatin which are present, they start gradually separated from the nuclear structures, and that will be released into the cytoplasmic content. So these are changes which are taking place during the prophase 2 stage. So this is a condition where this is related to one nucleus. In the 2 nucleus, the same type of changes will be observed. Coming to metaphase 2. During anaphase 2, all the chromosomes of both the nuclei will be released into the cytoplasm. So here now the spindle fibers have been formed. These spindle fibers which have been formed are perpendicular to the spindle fibers which have been formed during metaphase 1. Here this meiosis 1 is meiosis 2 is equal to the mitotic phase, because the spindle fibers are formed which are discontinuous fibers, and centromere is present on the equatorial plate of the equatorial region. This is equatorial plate. So the spindle fibers which is formed is perpendicular to the spindle fiber formed during metaphase 1. Then anaphase 2. During anaphase 2, the contraction of the spindle fibers will be taking place, so that the centromere will split and daughter chromosomes are formed. Now each daughter chromosome contains one chromatid, one centromere, and one DNA molecule. So that can be represented like this. So the contraction of the spindle fibers, so that the chromatids are moving towards the opposite poles. So likewise, the daughter chromosomes which will be moving toward this side, this pole, these 2 daughters are moving to this side. Likewise, at the 4 corners we can see 4 groups of chromosomes which have been accumulated towards 4 corners of the cell. Then telophase 2. During telophase 2, the 4 daughter groups of chromosomes which have been formed, they reorganize the nucleolar membrane, the nucleolus, and the endoplasmic reticulum, Golgi complex, to form into 4 daughter nuclei. So by the end of meiosis 2, 4 daughter nuclei have been formed, but these 4 daughter nuclei are quite different from each other, because the sequence of genes present on the chromosomes is different from each other, and they are structurally, morphologically and genetically also quite different from each other. And they are not only different from each other, they are different from the parental chromosomal constituent part also. So here at the end of this telophase, telophase 2, we can see that 4 daughter chromosomes have been formed. Whatever the changes which have been taken place during meiosis 2 are similar to that of mitosis. That is the reason why. And one more thing, there is no change in the chromosome number, there is no change in the gene sequence. That is the reason why the meiosis 2 is equals to mitotic stage, and that is the reason why meiosis 2 is called as homeotypic division or equational division also. Then cytokinesis. After the completion of nuclear division, the cytoplasm will also divide. So there are two types are there. One is simultaneous and successive type of cytokinesis. In the simultaneous type, first the nuclear divisions will occur. Both the nuclear divisions will occur. That is, at last 4 nuclei are be formed. After formation of 4 nuclei, the cytokinesis will occur at a time. Whereas here, after meiosis 1, cytokinesis will occur, leading to formation of 2 daughter cells. Then after meiosis 2 will occur. There after meiosis 2, once again the cytokinesis will take place. That is, here in the simultaneous type, the nuclear division will take place twice, but cytokinesis will occur only once after the completion of meiosis 2 only. But in the successive type, after meiosis 1, the first cytokinesis will occur, then 2 cells will be formed, then after meiosis 2 will be taking place, thereafter once again the cytokinesis will occur. Likewise, this type of changes can be observed, but which type of cytokinesis will take place in the plant cells means it is like a cell plate method, as like that we have seen in the mitotic division. Then significance of meiosis. So significance here, it is useful for maintaining the chromosome, maintenance of same chromosome number from one generation to another generation. And here crossing over is taking place. Due to crossing over, the new recombination of genes is taking place. The recombination of genes may lead to the new characters. So if the process is taking place for many number of generations, the new species will be formed, leading to the evolution process. So these are the major significances related to the meiotic division.